Hi, good morning, students. We are now discussing the competent host for the transformation with recombinant DNA. हम एक कॉम्पिटेंट होस्ट का चुनाव करेंगे ताकि हम रिकॉम्बिनेंट डीएनए का ट्रांसफॉर्मेशन उस सेल का करवा सकें। ट्रांसफॉर्मेशन की बात अगर हम करते हैं तो ट्रांसफॉर्मेशन इज a process by which a cell takes up naked DNA fragment from the environment, incorporates it into its own chromosomal DNA, and finally expresses the trait controlled by the incoming DNA. The tools described earlier will result in generation of recombinant DNA molecule in the laboratory. Finally, the propagation of these DNA molecules must occur inside the living cell or the host cell. Many kinds of host cell including E. coli, yeast, animal and plant cells are available for genetic engineering, and the kind of host cell to be used mainly depends on the aim of cloning experiment. For the expression of some eukaryotic protein, eukaryotic cells may be preferred host. Yeasts have been used extensively for functional expression of eukaryotic genes because they offer several advantages. Yeasts are simplest eukaryotic organisms like bacteria. Uh, in fact, yeasts are the simplest eukaryotic organism, and bacteria are single-celled, genetically well characterized, easy to grow and manipulate. They can be grown readily in both a small culture vessel and large-scale bioreactors. Plant and animal cells may also be used as host in gene manipulation experiment and for protein expression either in tissue culture or as cell in the whole organism to create genetically modified animals and plants. सिंस डीएनए इज़ ए हाइड्रोफिलिक मॉलिक्यूल, ये बहुत इंपॉर्टेंट पॉइंट है यहाँ कि डीएनए जो है वह एक हाइड्रोफिलिक मॉलिक्यूल है और इसकी एंट्री थ्रू सेल मेम्ब्रेन पॉसिबल नहीं हो सकती. सो बैक्टीरियल सेल इज़ must be made capable of taking DNA, and it is done by treating them with a specific concentration of divalent cations such as calcium ion with which increases the efficiency for the entry of DNA into the bacterium through the pores in the cell wall. Recombinant DNA can be forced in such cells by incubating the cell with recombinant DNA on ice. Recombinant DNA